Tutorial, I'm going to talk about a fruits example which has actually been published, and it was published in this publication, and I will place in the description of this video the link where you can download this publication. And this is interesting because actually in this publication the performances of two fruits models are compared, a weighted model and a non-weighted model. Now, the goal of this research was to quantify the contribution of two types of animal foods, terrestrial and marine, towards human diets. And in terms of proxies, there were two. We had carbon and nitrogen isotopic values measured in the collagen, extracted from bone from these humans. These food groups only had one fraction. Only one fraction was considered, I should say, and that was protein. And so the consumer isotopic signals were fully determined by the source protein, so this is a non-weighted model. And in terms of the offsets that were employed, well, in the case of the carbon isotopes measured in collagen, it was assumed that there was an offset of 5.0 towards the source protein with an uncertainty of 2.3, and in the case of the nitrogen isotopes, one offset of 5.5 with an uncertainty of 0.5 towards the source protein. Now, this was a concentration independent model. Okay, so I have this model here already prepared, and actually in the publication, 9 different individuals were modeled, but here I'm going to show you the outcome just for one. So changing this to 1 and then pressing enter, apply. And just to go through the different model options and parameter values. So this is a non-weighted model and concentration independent, but we do have the offsets. Okay, the two proxies, so that's the carbon and nitrogen isotopic signals measured in bone collagen, and then here we have the consumer data for this particular consumer, the two food groups, terrestrial and marine, only one food fraction protein, these offsets that I discussed previously, and the signal is fully determined by the protein. The protein values in the different food groups for carbon and nitrogen, and this is a concentration independent, so food sets these values to 100. So now we can generate the estimates, and you will see that the diet of this individual is predominantly terrestrial diet. You can see it here, okay, in the box plots, also in the probability distributions. I can show this as a smooth area, okay. And here we have the results report, so 94% plus minus 5 of contribution from terrestrial animals, and 6 plus minus 5 from marine. Estimates on fraction contribution, well, there's just one fraction, that's protein, so that's of course 100%. And the contributions from the sources towards the different signals, towards the carbon signal, nitrogen signal, well, they match exactly the main contribution, because this is a non-weighted concentration independent model, so they have to be the same. Now, as I said, there was also a weighted version of this model, and this is what I'm going to show you next. Okay, and in this weighted version, here it is, again the same two food groups. Now this is wrong, I have to correct here something. Okay, and now we can put this back. So, as I said, so two food groups, terrestrial and marine, as before. But now we are using a weighted model. So, in the case of the nitrogen measured in collagen, the nitrogen isotopes, it's still fully determined by the protein, but now we have a second fraction, carbohydrates and lipids. And the consumer isotopic signal for carbon is being derived in about 74% from source protein, but there's also a significant contribution, smaller but significant, coming from carbohydrates and lipids of 26%. Okay, and the motivation from this is actually this other publication, in which results from this publication is actually a meta-analysis of results from feeding experiments on omnivorous mammals, and using carbon isotopes as a tracer. And so from this meta-analysis, this is where I reached these values here. Okay. And the offsets between consumer carbon isotopic signals in collagen towards the source, in this case, is 4.8 plus minus 0.5, and in the case of nitrogen isotopes, it's 5.5 plus minus 0.5. Okay, and I have this model here, this one here. Okay, and again the 9 consumers, so I'm just going to model for the first one, pressing enter, apply, and let's go through the data. Now this is, well, I forgot to mention this, it's a concentration dependent model now. It's a weight model, and it's of course still an offset dependent model. We have the two same proxies, the same consumer data, the same food groups, but now we have two fractions. And here I mentioned protein and energy. Well, this means carbohydrates combined with lipids. Offsets and weights. Notice nitrogen is fully determined by protein, that's 100%. But now for carbon, it's 74% from protein and 74, and the remaining 26% from carbohydrates and lipids. That's the energy. Food values. Now, nitrogen values are the same as before, and here now we have not only the carbon isotopic values for protein, for both the marine and the for both the terrestrial and the marine food groups, but also the carbohydrates and lipid signals. In this case, since it's animal products, this is essentially the lipid signal for these animal foods. Okay, so this is for the terrestrial food group, and this is for the marine food group. Okay, and then the uncertainties. And then there are also concentration values for protein in these food groups and for the carbohydrates and lipids. And now we press estimate and give it a few seconds. And there will be more information provided by this model. So this model informs us, again, the main estimates here, and they're actually quite similar to the previous one. So this is 96 plus minus 3 and 4 plus minus 3. But now we also have the contribution of protein versus lipids. Let's say it like this, carbohydrates slash lipids, but being animal foods, it will be mainly lipids. And then we have the contribution of the sources towards the carbon signal and the nitrogen signal. And this nitrogen signal is only derived from the protein, so this is the contribution of protein coming from each food. And it doesn't match the contribution coming from each food towards carbon, because now the source of carbon in the consumer also includes the contribution from a fraction other than protein, that is the carbohydrates slash lipids. So that's why these numbers now are slightly different also towards this one, okay? Because now we have a weighted model. Anyway, I wanted to show you these two very simple models so that you can learn more about them and perhaps apply them to your specific problem. However, one thing that I would like to emphasize that it's very important that you define these numbers for your own problem. So although I'm using these numbers to address my specific problem, I think that it's important that each user finds values that are well suited to their specific problem. And also you could have other data that suggests that numbers different from these ones should be employed, and well, different offsets. Perhaps you prefer a weighted model or a non-weighted model, and perhaps the numbers that you have for these contributions are different. That's perfectly fine. Fruits can handle that without any problem, because one of the main aspects of fruits is its flexibility. So it's not linked to any specific values, and these should really be defined by the user after the user does a proper review of the literature and of the data that he has access to, and so he's able to define the numbers that fit the best to his specific problem. So, well, I think I don't have anything more to say, and well, that is it.